Hello, and thank you for joining me today. I sincerely appreciate your time. Today, I'm going to present a solution that I developed and implemented during my research work at UC Berkeley in an area of focus that resonates deeply with me on a personal level. In 2012, as I was continuing my previous 12-year career in enterprise architecture, I wanted to leverage my background to do everything that I could to help assist in ending childhood illnesses. From that time on, I have continued my career in enterprise architecture, but also strive to learn as much as possible in the area that is now referred to as bioinformatics, along with healthcare and life sciences. Throughout my career, I've been fortunate to work in many different business sectors to leverage emerging technologies to help solve some of the most challenging scenarios. The dream that I had in 2012 led me down the path of accelerating human genome sequencing with a focus on precision medicine. We are in very promising times to be able to leverage technology to meet the needs of our most complex challenges. In this presentation, I'm going to run through the solution that I developed for the most common form of pediatric brain cancer, medulloblastoma. Our focus will be on the intriguing realm of genomics secondary analysis, a critical process that helps us make sense of raw genomic data by aligning it with a reference genome and pinpointing variants. We'll be navigating the robust infrastructure of Amazon Web Services, or AWS. We'll work with sequenced reference and patient genomes, which are stored as fastq.gz files. These files will be uploaded into an AWS S3 Bucket, setting the stage for our analysis. Our next step involves crafting genomics analysis Docker images, which will be deployed into AWS Elastic Container Registry Repositories. Subsequently, we'll establish an AWS Fargate Compute Environment and an AWS batch queue responsible for orchestrating the running of our Docker images on an AWS Elastic Container Service. As we progress, we'll assemble AWS batch job definitions for each of our 3 genomics analysis toolsets. This will be followed by the creation of AWS step functions for a state machine responsible for running an orchestration workflow. The end goal is to generate variant call format, or VCF files. These files are vital in analyzing and identifying actionable mutations, enabling the application of therapies targeted at inhibiting these specific mutations. We're living in an area of unprecedented advancements in healthcare and life sciences, where the promise of developing precise patient-specific treatments is not just a dream, but a tangible reality. Let's explore this exciting journey together. We begin the process of genomic sequencing with the collection of bacterial cells. DNA is extracted from these cells and subsequently fragmented. This fragmented DNA undergoes amplification through polymerase chain reaction, or PCR. A sequencer then identifies the nucleotide combinations in these fragments. The result is a set of fastq.gz files that contain sequenced genomic data ready for deeper analysis. Next, we transfer the sequenced genomic data into an AWS S3 Bucket. These uploaded fastq files are a representation of our DNA sequences. Next, we'll construct Docker files for our integral bioinformatics tools, BWAMem, SAMTools, and BCFTools. But first, let's briefly understand what these tools do. BWAMem is a software package for mapping low-divergent sequences against a large reference genome. It's effective in aligning sequence reads of various lengths, making it an essential tool in our genomic analysis. SAMTools provides a suite of utilities for interacting with and post-processing short DNA sequence read alignments in the SAM, BAM, and CRAM formats. It's a vital tool for manipulating alignments, including sorting, merging, indexing, and generating alignments in a per-position format. BCFTools is designed for variant calling and manipulating VCFs and BCFs. It plays a critical role in handling files containing information about genetic variants discovered in a genome. Now, with a better understanding of our tools, we'll create Docker images from these Docker files. These images will be uploaded to the AWS S3 Bucket, setting the stage for our subsequent steps in the genomic analysis. In our next step, we'll deploy the Docker images to AWS Elastic Container Registry Repositories using command line utilities. It's crucial to note that each tool within the AWS ECR repository has a distinct Uniform Resource Identifier, or URI. This URI plays a pivotal role as it will be referenced within the JSON file during the subsequent AWS step function state machine creation step. We kickstart the AWS Fargate Compute Environment, which serves as our central hub for managing and allocating computational resources. AWS Fargate is instrumental in its ability to run containers directly without having to manage the underlying EC2 instances. The choice of the appropriate virtual private cloud, or VPC, subnets, and security groups is a crucial step in this process. It's also worth mentioning the significant roles played by Fargate spot capacity and virtual CPUs, both of which are instrumental in optimizing our resource utilization. The creation of the AWS batch queue is the next step. It plays a crucial role in job scheduling. The Amazon resource name, or ARN, for the queue is needed during the subsequent AWS step function state machine workflow, start execution, and output to AWS S3 step. We create AWS batch job definitions for BWAMem, SAMTools, and BCFTools. This step involves the selection of the orchestration type, job role, configuration, vCPU count, memory amount, and Docker daemon volume addition. The creation of the AWS step function state machine involves importing the definition JSON file, which includes the Uniform Resource Identifier, or URI, that we collected during the previous pushing BWAMem, SAMTools, and BCFTools Docker images to the AWS ECR Repository step. The state machine design is displayed, and we enter the state machine name and select or create the permissions execution role. We begin our workflow by preparing resources and inputs, leveraging AWS Fargate to load Docker images. We then use the BWAMem algorithm to align sequence reads to our reference genome, creating a SAM file. This file is transformed into a sorted index BAM format using SAMTools, allowing us faster data access. We call variants per chromosome to enhance manageability, then utilize BCFTools to review aligned reads coverage and call variants on the reference genome, resulting in a VCF file of identified variants. Finally, we start the execution of the AWS step function state machine batch workflow. This involves importing a JSON file which includes the job queue Amazon resource name, or ARN, from the previous creating the AWS batch queue step, the AWS S3 source, and output folders, along with the chromosomes of focus for the analysis. At the end of the workflow, the chromosome VCF output files are stored in the referenced AWS S3 output folder. Let's imagine our DNA as a huge instruction manual that controls how our bodies work. Sometimes there are typos, variations, or mutations in this manual that can cause diseases like pediatric medulloblastoma. A VCF file is like a highlighter that marks these variations in the manual for each chromosome of focus. As we have laid out this solution, we can read these highlighted sections and identify which mutations are causing the cancer to grow, which are actionable mutations. Once we know what's driving the disease, scientists and oncologists can design very specific precision treatments to correct or manage these variations. This is like creating a custom-made plan to fix the typos in the manual. Using the information from VCF files, especially chromosomes 2, 3, 7, 8, 9, 10, and 17 in this example, we can develop therapies that are precisely targeted to a patient's unique genetic variations, offering a personalized treatment approach with less toxicity and greater efficacy. What excites me most about the continued evolution of technology is the ability to demonstrate the humanistic side of the complex challenges that these toolsets can help solve. There are limitless capabilities that we have across many different business sectors. It was wonderful speaking with you today, and I thank you for your time. Please feel free to reach me if you have any questions at all.